I'm Janet Smith, and my lab studies the 3D structure of proteins in order to understand how they function. Proteins are not only the structural components of our muscles and other tissues, they also do a lot of the work that goes on inside living systems, including us. We use X-ray crystallography to determine protein structures because we mostly want exquisite detail. So what we do is isolate and purify the protein molecules that we're interested in, and we line them all up in a crystal. We scatter X-rays off of them. They all diffract in phase, and so we can reconstruct the structure of what was in the crystal. We have a molecular understanding of the inner workings of a cell if we can see the structure in detail of proteins. It's kind of like the state of Michigan. You go like this, and there's Michigan. Well, that's not very detailed. We want the atomic structure. One of the proteins my lab studies is the flavivirus NS1, and this is an important protein that helps virus be more pathogenic. It is toxic, it also assists the virus in hiding from the immune system, and it makes the virus more infective. Flaviviruses include some really serious human pathogens, and there are at least 5 dozen flaviviruses. The biggest public health threat internationally is dengue virus, which infects hundreds of millions of people around the world every year, sometimes fatally. We and the whole community did not have a hypothesis for how it does what it does. We needed to see it. So we made the protein, we crystallized the protein, we got its 3D structure. We were able then to go back into the literature of all the things that were known about the viral life cycle and figure out which part does what thing. The available vaccines for dengue virus don't work equally well on all strains of dengue. NS1 could provide a much more general vaccine for example, dengue virus. There are many tools that we need in order to develop drugs. Knowing the structure is an essential one of those tools. You can't design a drug if you don't know where it's going and what it's interacting with.